Yeah, hi everybody. This is and it's relatively brief. The last portion is about the respiratory. What I sent you previously, effectively, it was the same, similar material, just maybe with a slightly accelerated degree of difficulty. I gave it to my anatomy and physiology people, covering immune and blood vessels. So you've already got that information. It didn't change, which leaves us with this. So I will go over here and open up that particular screen so I can tell you. So one I did start to talk about with them because I was looking at your exam, not their exam, was the interesting thing about respiration with regard to resistance. The resistance is interesting if you look at the curve on the PowerPoint sequence. It increases initially as you go from bronchi, the large main bronchi, to the medium-sized bronchi when we begin to get to like tertiary or segmental bronchi that are there. That's where resistance increases because it's a function of diameter. Okay, not individual diameter, but total diameter. So you have your diameter starting to decrease, but you don't have that many branches. And then interestingly, it begins to increase dramatically, even though the passageways get smaller. There are so many more orders of branching that resistance decreases dramatically if you look at the curve. So that's one of the first points I just wanted to amplify since I had commented on it earlier. And again, there's not a whole lot. There's probably this will take 5 minutes. This whole thing. What's going on here? Let me go here. Here we are. Okay, neat stuff. What's the role of surfactant? Obviously, it's about surface tension. If we decrease surface tension, it is much more easily. It is much less likely to be a better way to express it that there would be adherence of the individual alveolar cells. That's the biggest problem, is being able to reinflate something after things stick together. Surfactant is produced roughly at 26 weeks of gestation and continues to be produced through your lifetime. Fortunately, now we have, pardon me, doing this first thing in the morning. Fortunately, now we have surfactant that is manufactured and produced, so we can introduce this in preemies, roughly age 20, 20, 23, 24 weeks of gestation, with a reasonable chance of survival. So it's been quite a significant feature. Remember, it comes from those type 2 cells. The type 1 cell is the standard alveolar simple squamous epithelia. The type 2 cell is more of a glandular type of a cell in the sense that it produces the surfactant, that particular secretion. And the other cells that are there are those macrophages, which are just in very, very large numbers and effectively constantly trying to maintain the I hate to say cleanliness, but the relative sterility of all of the alveoli. Years ago, we used to think that this was a sterile area. Now it is not. Now we know that there's occasional particles that come down with some degree of frequency, and that's what those macrophages do. So that's significant. Now we look at the exchange of gases. Okay, effectively, this is the model for simple diffusion: oxygen and carbon dioxide. The big difference is that their concentration gradients based on partial pressure of gases are different. Okay, not only different enough to create a concentration gradient, but relatively different in the sense that oxygen relies on concentration gradient, much less so with carbon dioxide. Even though they cross equally, it's because of the membrane solubility, which is much greater for carbon dioxide. It's just more compatible. That's there. And remember, effectively, both of these, to a tiny extent, much less with oxygen, much more so with carbon dioxide. We have gas that's dissolved. Of course, the bulk of carbon dioxide is not dissolved gas, but carbon dioxide that has the aid of carbonic anhydrase and water have made carbonic acid. Uh, the bulk of the oxygen, of course, rides on the heme molecule, okay, so to speak. There's a limited amount, probably, depending on the textbook, of roughly 20% of carbon dioxide that travels on the globin, a limited amount that dissolves in gas, and then obviously about 70% that utilizes the carbonic acid buffering system and the aid of carbonic anhydrase to effectively dissolve in the plasma. So that, remembering those modes of travel, certainly remembering the uniqueness and the shape changes associated, really indicated by that oxygen dissociation curve, the ability for this magical protein, we call it a metalloprotein molecule because of the iron, the heme unit associated with the globin, to change shape as it's loading and it's unloading. When it unloads, it changes shape to make it easier to unload. When it loads, it changes shape to make it easier to load. So I think that's really important as well. Remember that respiration, the driving force for respiration, and erroneously people thought it was about oxygen. Really, if oxygen, you're relying on that to drive respiration, you're in big trouble. It's basically carbon dioxide, partial pressure of carbon dioxide or percentage of carbon dioxide, followed by pH, because obviously with the carbonic acid buffering system that's involved, that plays a pivotal role in pH, and then oxygen concentration. So remember that sort of batting order that's there. The distinction between obstructive and conductive diseases. That's really a reason for doing the lab with the pulmonary function studies. The conducting division is all about moving air in and out, relying on pressure changes. When that is inefficient, when that FEV at 1 second falls below that 75 to 85% range, at that point we have an impairment in the conducting division, and we call that an obstructive disease. By the same token, if we calculate vital capacity, and if we see a diminishing number of vital capacity as would be expected given body type parameters, history, etc., then we know that that's an impairment of the exchange or respiratory division. Okay, so that now we're looking at different. So it's not a conductive portion, it's the exchange division. So we have an impairment there, and that means a limitation of oxygen and carbon dioxide to exchange because we don't have enough functional alveolar space to do so. We have a diminishing amount of functional lung tissue, we want to think of it this way. So remember the different volumes and capacities. We have the tidal volume, the sort of the in and out without forcing it whatsoever, about a half a liter. We have a larger inspiratory reserve and an expiratory reserve. We add the three of those together. We get vital capacity. The total lung capacity is that number plus adding what we call the residual volume, which includes dead space and a variety of other factors that are there. So remember, when we don't have enough vital capacity, the fancy term is called a restrictive disorder, which is affecting the exchange division, and an obstructive disorder affecting the conducting division. Look up the POR, BOHR, and Haldane effects to see which one of those has to do with unloading oxygen, which one plays a role in carbon dioxide. Are they similar? Are they different? Etc. That's there. Remember, breathing is effectively effortless. The only time when we have to use muscular activity, and it's minimal, comparatively speaking, other than causing our diaphragm to flatten, which is an active process, contracting it effectively, and our external intercostals. Everything else is just relaxing them, and everything else takes place because of elasticity and just muscle relaxation. And we let the pressure do the rest of the work. We've increased flow in, and when we relax them, then that facilitates, then the difference between atmospheric pressure and lung pressure lessening, that whole volume and pressure are constant, are important. And I think all of those are the factors that you have to know. Everything else, the definition of emphysema is important. Effectively, there's a limited number of respiratory questions. To summarize, I'd say of this 55 questions, 30 to 35 are immune system, and maybe 8 to 10 apiece are from respiratory and blood vessels, and that summarizes it. Like I said, there wasn't a whole lot. Sorry for the delay in it, just so much stuff going on. Glad to get it done, and I will send this to you post haste. Goodbye.